Hello and welcome back to educator.com and to biochemistry. So we've been talking about lipids for the past couple of lessons, and today what we're going to do is close out our discussion of lipids by talking about what I call the biologically active lipids. That's not to imply that the other lipids are not biologically active, but what I mean by that is that these lipids that we're going to talk about today, they are not used as fuel storage, in the fat cells or sequestered in membranes. So in some sense, they're sort of locked in, not really moving around much. By biologically active, I mean something that's actually free to move around and engage in some sort of chemistry. So that's all I meant by that. I didn't mean to imply that others were not biologically active. Okay, so let's go ahead and get started. So let's see. Let's go ahead and start by discussing a molecule called phosphatidyl inositol. So let me go ahead and draw a structure here. Let me see, where should I draw it? And let me go ahead and use blue. I always like blue. So let's go this way. Let's go a little bit further over here. So C, C, C. Let's go, we have our CH3, 10, CH3. And then we have our O, and then we have our other carbonyl here. And then we have CH2, 7. Then we have another C. We have a double bond here. And then we have a CH2. It looks like we have another 7 and CH3. So this is our diacyl glycerol part. So again, we're just attaching something to glycerol. And of course, well, not of course, in this particular case, we are going to have a glycerophosphate. So glycerol, we're going to have the phosphate linkage. So we'll go ahead and put that in, and we have our oxygen. So this is a phosphate ester. And let's go ahead and draw in our, what looks like a sugar unit. So this is going to be well, I'll go ahead and draw things in, and then I'll go ahead and label. So that's that, that's that, that's that, and that's that. And I'll go ahead and label these. This is 1, 2, 3, 4, 5, and 6. In this particular case, this is the numbering on this particular section. So this is phosphatidyl inositol. So or phosphatidyl, however you want to pronounce it, inositol, and it is a glycerophosphate or a phosphoglycer, whatever. It's glycerin backbone. It contains a phosphate. That's what's important. All these names make me crazy. They always have. I mean, they're kind of cool in the beginning, but eventually they start to wear on you. So a glycerophosphate. Let's just go ahead and call it that. Okay, so something really, really interesting happens here. Now, when this molecule, is activated, so this is an example of its biological activity, is activated by an extracellular stimulus, so something outside the cell binds and activates it, gets this process going. Stimulus, so extracellular, the following reactions take place. The following, I'll just go ahead and write reactions, not a problem. The following reaction sequence, how's that? Okay, so the following reaction sequence takes place. Okay. Let me go ahead. I wonder if I should start on this page or actually go to the next page. You know what? Let me just go ahead and start on this page. That's not a problem. So let me go back to blue. So I'm just going to write phosphatidyl inositol. Okay, so this is that. So this is in the membrane. This is a glycerophosphate. So this is one of the real quickly on the side here. So you had your, it's real tiny. So you had this, right? And then you had your phosphate bond. It goes on. So it's going to break that bond, the glycerol phosphate bond. Okay. Actually, it's going to, well, actually, that's not true. It's going to end up breaking this one. It's going to end up breaking this one. So water is going to hydrolyze it. So water is going to come and attach to that. So phospholipase C is the membrane. It hydrolyzes the glycerol phosphate bond. So it's actually water that is coming in, and it separates this into two separate molecules. The first molecule that it separates it into is IP3, and this is called inositol triphosphate. Inositol triphosphate. So it is just the six-membered ring plus the phosphate and the two other phosphates that are attached to it. So inositol triphosphate. Okay, so this IP3 is released into the cytosol. Remember we said it's on the inner leaflet, so once it breaks that, the IP3 molecule just floats into the cytosol. I'll show you a picture of it in just a minute, so don't worry about that. Is released into the cytosol. Okay, and the other, of course, is the diacylglycerol. The diacylglycerol that it stays in the membrane. It stays in, well, I don't need to put parentheses around it, that's fine. So the diacylglycerol part, it stays in the inner leaflet of the membrane, stays in the membrane. Okay, now here's what happens. This IP3 that's now in the cytosol, it causes the release of calcium ion from the endoplasmic reticulum, from the ER. I'll just put ER for endoplasmic reticulum. Now, the calcium ion and the diacylglycerol that's still attached to the membrane, they activate together. The calcium and the diacylglycerol. They activate an enzyme. They activate, well, we'll just give the name here, protein kinase C, and we just call that PKC, very, very important enzyme. So PKC, protein kinase C, it begins its function of regulating further enzymes by phosphorylation. So let's just recap, and then we'll go ahead and take a picture of it. So you have this phosphatidyl inositol, an extra cellular stimulus, ends up causing it to be phosphorylated. It ends up breaking it up. It phosphorylates it. And so what you end up with is this phosphorylase, phosphatidyl inositol 4,5-biphosphate, the PIP2. Phospholipase C in the membrane, it hydrolyzes the glycerol phosphate bond. It breaks it up into two separate things. The IP3 is released into the cytosol. The diacylglycerol stays in the membrane. The IP3 causes the release of calcium from the endoplasmic reticulum. The calcium and the diacylglycerol activate protein kinase C. And protein kinase C starts its function of regulating further enzymes to do whatever it is that they're going to do. So this signal pathway that's taking place from this phosphatidyl inositol. Okay, now let's go ahead and take a look at this schematically or diagrammatically here. Okay, so here's what's happening. So what we've done is we have this phosphatidyl inositol, and it's going to end up being phosphorylated. So now it's going to be PIP2, right? So we have our PIP2. So this is extracellular. This is outside the cell. This is inside the cell. This is the membrane right here in green. Go ahead and go to, that's fine. I'll go ahead and leave it as red. Now here's what happens. So the phospholipase C actually breaks off the inositol part, the polar part, and it releases it into the cytosol. Okay? This IP3 interacts with a receptor, the endoplasmic reticulum, and it causes calcium to be pumped out into the cytosol. The calcium binds to protein kinase C. Protein kinase C binds to the diacylglycerol, which was still in the membrane, and together, that combination, it starts a new series of events. It starts phosphorylating enzymes which go and do whatever it is that they're going to do. So this phosphatidyl inositol serves, it's a biologically active lipid. It serves as basically starting a series of signals to go on and do whatever it is that the cell needs to do. So that's it. Okay. Now let's move on. Let's go back to blue here. Now let's talk about another class of biologically active lipids. They're called the eicosanoids. And what they are is paracrine hormones, and they act only on cells near, so that's what paracrine means. Paracrine means they act very near the place where they're actually made. They're not actually sent, they're not pumped into the blood and sent to different parts of the body like other hormones. So this is a paracrine hormone. It acts very, very locally. That act only on cells near the region of hormone synthesis. Let's just go ahead and make sure we write all this out. Hormone synthesis and not transported in the blood to remote tissues. So in general, when we think of hormones, that's what we're thinking about. We're thinking about hormones that are pumped into the blood and sent out to other parts of the body to do whatever it is that they need to do. Remote tissues and cells. Okay. Now, let's write this one in red. Their effects are both very diverse and very profound. So these eicosanoid hormones, there are lots of things that they do, not just one or two things that they do, lots of different things that they do. And when they do them very, very profoundly. The effect is very, very deep. So it's not these are not mild hormones by any means. And very profound. And let's see what else. They are derived, all of them, they are derived from something called arachidonic acid. Arachidonic acid. Okay. And we'll be showing you some pictures in just a minute. And let me go back to blue for this one. So, membrane phospholipids. Phospholipids. So arachidonic acid is just it's a fatty acid. That's all it is. It just has a certain length carbon, has some double bonds in it, but it's just a fatty acid. So as a fatty acid, it can participate in reacting with glycerol to form some glycerophosphate that's in the membrane. So membrane phospholipids containing the arachidonic acid are hydrolyzed when needed, are hydrolyzed by an enzyme called phospholipase, A2 to be exact, to release arachidonate. And remember, when we say arachidonate, it just means it's the ionized form. Arachidonic acid is protonated, arachidonate deprotonated. Okay, so that's what happens. So these eicosanoids are paracrine hormones. They act only on cells near the region of hormone synthesis, and they're not transported in the blood to remote tissues and cells. Their effects are diverse and profound. They're all derived from arachidonic acid, and the arachidonic acid comes from the membrane phospholipids that contain it and are hydrolyzed when needed. Okay, so let's go ahead and take a look here, see what we've got. So this is some membrane phospholipid, and so notice our glycerol. So let's go ahead and mark off the glycerol part. That's 1 carbon, that's 2 carbons, that's 3 carbons. So on one of them we have, of course, our phosphate group, and of course we have this is our polar head group, okay? So this is, looks like choline here. And then, of course, we have this one. And here, of course, we have the ester linkage, our first fatty acid, diacylglycerol. Here is one of them. Here is the other one. In this particular case, this is our arachidonic acid right here. It's attached in ester linkage to the glycerol. So what it does, it actually hydrolyzes that. So it breaks this, and it ends up creating our arachidonic acid. So this right here is a membrane lipid containing the arachidonic acid. And so this is our arachidonic acid right here. And of course, once we break that, this is our arachidonic acid, our fatty acid. So this is arachidonic acid, or arachidonate. It doesn't really matter. Arachidonate just means you've deprotonated that, and now you have the negative charge on it. So that's it. 1, 2, 3, 4, 5, 8, 11, 12, 13, 14. So that's it. 4 double bonds. So 1, 2, 14, 15, 16, 17, 18, 19, 20. So this would be 20 carbons. There are 4 double bonds, and you have a delta 5, 8, what do we say, 11, 14? Yes, 11, 14. So that would be the delta notation for arachidonic acid. That's it. Okay, so now let's talk about the classes of the eicosanoids. So there are 3 classes of eicosanoids. You have the prostaglandins, you have thromboxanes, and you have the leukotrienes. Okay, so let's take a look at some of these and see what we can do. And we'll talk a little bit about each one. So prostaglandins, thromboxanes, and leukotrienes, all of them are eicosanoids. They serve different purposes. So let's see what we've got. Okay, so we've got arachidonic acid. So I just wanted to put it up there for you to see it again. So let me just write here. So arachidonic acid. So from arachidonic acid, we end up forming, so let's see what we've got here. So we have, well, let me just go ahead and write out what the names of these are. So here we have, this is a prostaglandin. This happens to be prostaglandin A1, I think. Yes, A1. And notice, well, let me just write out. And then over here we have the thromboxane. And then I'll go ahead and list some properties for each of these. And here we have the leukotriene. Leukotriene. And of course, triene. Yes, you've got your conjugated triple bond, which is one of the characteristics. This happens to be leukotriene B4, and this is thromboxane A2. Okay. Well, it's not well, that's fine. We can just write it. Yeah, that's fine. I'll go ahead and write it like that. Okay, so now let's talk about some of these things. So let's start off with our prostaglandins. Let me go back to black here. So prostaglandins. Okay, so an example of a prostaglandin, an example of a thromboxane, and an example of a leukotriene, and of course there are characteristic features here. So the prostaglandins, they contain a 5-membered ring. They contain a 5-membered ring. There's your 5-membered ring. Okay. There were 2 groups originally. Called originally the PGEs and the PGFs. So the prostaglandin E series, the prostaglandin F series. Now, there are other groups. There are others, like the one above. This is the prostaglandin A series, prostaglandin A1, prostaglandin A2, A3, whatever, just like prostaglandin E1, E2, E3, prostaglandin F1, F2, F3, that's it. So PGE, PGF, PGA, and so on and so forth as more and more get discovered, I guess. And there are others. Let me see. Now there are others. Let's see, what else can we say about prostaglandins? Okay, let's talk a little bit about what they do. Prostaglandins often regulate the synthesis. So again, a lot of these molecules, they don't just do one thing directly. They can, but often what they do is they start a process, and that process has several different steps down the line. So you can speak of it doing this, but what it ultimately does is maybe this. So in this particular case, prostaglandins, they often regulate the synthesis of 3', 5' cyclic adenosine monophosphate, an intracellular cellular secondary messenger. Secondary messenger. And what we mean by secondary messenger is, let's say from outside the cell there is a primary messenger, some sort of a molecule that binds to the outside of the cell. Something happens and it signals the inside of the cell. The inside of the cell produces 3', 5' cyclic adenosine monophosphate that is mediated by the production of some prostaglandin, and then this 3', CMP goes on to do what it does. So it's a secondary messenger. It's down the line a little bit. Tertiary messenger, quaternary messenger, things like that. Okay, let me go ahead and draw the structure real quickly of this thing. So we've got our, let's see here, just so you know what it looks like. Boom, 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 boom. Okay, we have our OH. We have our O. Let's go ahead and put the phosphate here. Let's go ahead and put, so this is adenine. And you remember the adenine itself takes the normal numbers. The sugar takes the prime numbers. So this is 1', 2', 3'. This is 4', and this is the CH2. This is 5'. So we've got O. That's that. And let me go ahead and put the double bond here, and I'll go ahead and do that. So this is cyclic adenosine monophosphate, 3', 5', if you want to be specific about where it's connected. So that's it. That's prostaglandins. Okay, so let's do some information about thromboxanes. So let's go ahead and do this in blue. So we have our thromboxanes. Now for these, we have a 6-membered ring. They have 6-membered rings containing an ether. Containing an ether. Okay. Okay, they are synthesized by platelets and are involved in blood clotting, hence their name, thromboxane, thromb. Okay, and this one is definitely good to know, NSAIDs, okay, such as aspirin and ibuprofen. NSAID, a non-steroidal anti-inflammatory drug. An aspirin is an NSAID, an ibuprofen is an NSAID. These things, they block the synthesis of prostaglandins and thromboxanes. By inhibiting the enzyme prostaglandin. Actually, I'm going to go ahead and just write the enzyme. Yeah, I'll go ahead and write it out. That's not a problem. Prostaglandin H2 synthase better known as cyclooxygenase. God, these names just longer and longer and longer. Also called COX. So these non-steroidal anti-inflammatories like aspirin, like ibuprofen, and there are several others, they block the synthesis of the prostaglandins and the thromboxanes. They inhibit an enzyme called prostaglandin H2 synthase, otherwise known as cyclooxygenase. This is an enzyme that you're going to talk about a lot for those of you that go on in biomedicine. Okay, so let's say a couple of words about the leukotrienes here. Let's see, do I have another page available? I do, so you know what I'm going to do? I think I'll go ahead and, it's fine. Let's go ahead and do this one in red. So now let's say a couple of words about the leukotrienes. The leukotrienes, they happen to contain 3 conjugated double bonds. So if you go back to that picture, you'll see that 3 of the bonds, they might have more double bonds than that. Like in the picture, I think we had 4 double bonds, but 3 of them are going to be conjugated. They're going to be one after the other. Not directly after the other. It's going to be double, single, double, single, double, single, that way. That's what conjugated means. There's always a single bond between them. So it contains 3 conjugated double bonds. That is a characteristic feature. That's the triene part. But that doesn't mean it has only 3. So it might have more. It might have only 3, but it might have more. So 3 conjugated double bonds. And let's see. They are involved in inflammatory responses. You know what? Let me just go to the next page. Let's see. Okay. In particular, leukotriene D4 is responsible for constriction of the airway during an asthma attack, or during anaphylactic shock or anaphylaxis. Let's go ahead and write it that way. Oh, spelling. Anaphylaxis. There we go. So this particular leukotriene, D4, that's what's responsible for, let's say, if you're allergic to nuts and your throat closes up, that's what does it. So there you go. Okay, now let's talk a little bit about steroid hormones. Let me see here. Do I have? Yes, I have them there. I wonder if I should do it on the next page. Yeah, you know what? Let me go ahead and actually write everything on the next page because we have the pictures on the next page. So let me go ahead and move over here. So let's talk about the steroid hormones. So this is another class of biologically active lipids. So they are lipids. They have certain biological activity as free molecules. So the steroid hormones. Now, they have the sterile ring structure, as you see, 1, 2, 3, 4, 1, 2, 3, 4, 6, 6, 6, 5, 6, 6, 6, 5, A, B, C, D. Remember, we talked about sterile ring structure. A ring structure, but lack the alkyl chain, on ring D. So you remember when we looked at the sterile structure, this is ring D right here, okay? Remember there was this long alkyl chain attached to it? So these have the basic structure, they have the 4 rings, but they lack that alkyl chain, and as you can see, they actually have other polar groups up here. So now they are highly oxidized, and reasonably polar. And again, I mean, mostly we have, you know, this hydrocarbon basic structure, but you see an oxygen here, the carbonyl group. You see the alcohol group over here, alcohol group over here, alcohol group over here. It's reasonably polar, at least on the 2 ends. So it's reasonably polar. Now, they move through the bloodstream attached to proteins, and they act on cells by entering them interacting with highly specific receptors, and when we mean highly specific, we're saying that the receptor recognizes just that particular molecule. Okay, receptors, and triggering changes in metabolism and gene expression. So these are very, very, very powerful hormones. I mean, they actually change gene expression. They affect what proteins are now going to be built to do whatever it is that proteins do. They change metabolism. They have deep, fundamental impact on what the body does. Okay, so over here we have, let's do this in red. So testosterone, the thing that essentially makes men men, with all of their masculine characteristics, is a steroid hormone. This is testosterone. Over here we have the estradiol, one of the primary hormones that makes women women, that gives them their particular characteristics. These are very, very similar. This should show you that there's actually very, very little difference between men and women. It's strictly a very, very small chemical difference. So essentially the same molecule for all practical purposes. There it is. So two very, very powerful steroid hormones. Obviously very powerful because as you go through puberty, men develop one set of characteristics, women develop another set of characteristics, and these are the hormones that induce those things. Okay, so now let's go ahead and finish off this discussion by talking about the fat-soluble vitamins. So let's see here. So our fat-soluble vitamins are, I don't know if I should write them. That's okay, I'll go ahead and write it over here. Okay, so our fat-soluble vitamins A, D, E, and K. So 4 vitamins that the body needs. They are fat-soluble vitamins. So let's go ahead and see. These are isoprenoids made by the condensation, just a fancy word for putting together, made by the condensation of multiple units of the molecule isoprene. And let's go ahead and just do a quick structure for isoprene. Double bond, C. I'll go ahead and put the H here, and then I'll go ahead and put another bond there, CH2, and I'll just go down below, that's fine. So this is isoprene, 1, 2, 3, 4, 5. When you polymerize this molecule, when you just put one isoprene unit and another isoprene unit and another isoprene unit, that's what that means. These isoprenoid vitamins, the A, D, E, and K, they're derived from isoprene, just a bunch of units. So they always come in increments of 5. So 5, 10, 15, 20, 25, 30, 35, 40. That's the number of carbon atoms that you're going to find in these particular vitamins. Okay, so this one over here, this is vitamin D3. I hope I have enough room to write a little bit about these things. So let's see. So this is, up here is vitamin D3, also called cholecalciferol. Now this is a prehormone. So in this particular form, it isn't really doing anything. Well, what ends up happening, so it's connected. So this is converted to 1,25 dihydroxy D3. Dihydroxy D3. So what we mean by that is one step in the kidney, one step in the liver. Each one of those steps attaches a hydroxy at the number 1 carbon, at the number 25 carbon. So now what you have is 1,25 dihydroxy cholecalciferol. This is the biologically active form of vitamin D3. Okay, and what this does is it regulates calcium uptake, calcium levels and uptake. So intimately involved in the incorporation of calcium into your teeth and bones. There's a lot about vitamin D that we actually do not know yet, and that's kind of the exciting part. So the field is wide open for how this thing does what it does, why it does what it does, and does it do other things. Really, very, very exciting. For all of these fat-soluble vitamins, we actually don't know very much about them at all. Okay, so now let's see here. So this is vitamin A. Let me go ahead and go to blue for this one. So I'm going to go ahead and just write what they are here, and on the next page I'm going to talk a little bit about them. So I wanted to do it on the same page, but that's not a problem. So this is our vitamin A. Okay, and this is our, I'll go ahead and do it over here. That's fine. This is vitamin E, and this is vitamin K. Okay, so now I'm going to go ahead and move to a new page and write a little bit about each. So let's talk a little bit about vitamin A. And after this, we'll go ahead and close off the discussion of the biologically active lipids. So it is a hormone. Okay. It also happens to be a visual pigment. Pigment of the vertebrate eye. Okay. The vertebrate eye. So when you see, it's vitamin C actually doing something with this protein called opsin. You'll discuss it later on, but it is what actually ends up sending the signals to your brain to let you know that you actually see and what it is that you see. So very, very important. It's also a pre-hormone. And its bioactive form as a hormone is retinoic acid. So when you look at the molecule, you had that, you had that hydroxy at the end. Well, this thing is converted to an aldehyde, and then it's converted again to a carboxylic acid. Everything else stays the same. When it's turned into the retinoic acid, that becomes the active form of the hormone that goes and does what it does. That's a different function from its function as a visual pigment. So it acts as a visual pigment in one form called retinal, the aldehyde form, and it's oxidized again to retinoic acid. It goes and does something else as, and it serves another hormonal function. Okay, so let's talk about vitamin E a little bit. Vitamin E, okay. They are called tocopherols. So when we talk about vitamin E, we're not talking about just one particular molecule. I gave you a picture of one particular molecule. There are slight changes in that, and that whole class of molecules that look like that have certain changes, those we call the tocopherols. So vitamin E is like a series of molecules that are very, very, very similar. And we call them the tocopherols, but we refer to it as vitamin E, so just so you know. And vitamin E is an antioxidant, a very, very powerful antioxidant. And antioxidants, they do one thing. They react with oxygen radicals. And you remember from organic chemistry, a radical is something that has just one electron instead of the two electrons, so they're a highly reactive species. Oxygen radicals, and other free radicals, not just oxygen radicals, and other free radicals to prevent the oxidation of membrane lipids. So, lipids are very, very prone to oxidation very, very easily. So what vitamin E does is it keeps them from oxidizing so that they can maintain the integrity of the cell. That's what it is. So it runs interference. These oxygen radicals and these other free radicals that are produced in, the normal course of metabolism, they're going to react with lipids and they're going to do all kinds of damage. Well, vitamin E prevents that by reacting with them so that they can't react with the lipids in the membranes. Okay, and now let's go ahead and say, you know what, why don't I go ahead and use the next page. A couple of words about vitamin K, and we should be done. So vitamin K, it is intimately involved with prothrombin. A blood plasma protein involved in, you guessed it, involved in blood clotting. So vitamin K, very, very important for blood clotting. And again, these fat-soluble vitamins, we know a lot about them, but we don't know a lot about them. So it's a very, very fertile area of research. And I could just imagine the things that we're going to be discovering over the next 10, 20 years for these particular vitamins. So thank you for joining us here at educator.com. And this closes out our discussion of lipids. Take care. See you soon. Bye-bye.